Thank
So
you.
let me introduce our third speaker for this second half of the session. He is Dr. Darwin Bandoy. Dr. Bandoy is an assistant professor at the UP College of Veterinary Medicine and a PhD candidate of Integrative Pathobiology at the leading veterinary school in the world, the School of Veterinary Medicine, University of California, Davis, as a Picari One Health Scholar. He graduated class valedictorian and cum laude from the UP College of Veterinary Medicine, registering the 10th highest GWA since 1908. He is a member of the UP COVID-19 pandemic response and one of the first to release pandemic models and epidemiological metrics for COVID-19 in the Philippines. As a response to the need for pandemic data, he initiated the creation of the Philippine Society for Epidemiology. So I will now give the screen to Dr. Bandoy.
So, good afternoon. Is the audio okay?
Yes, it's okay,
You're
Darwin.
okay.
Okay. So, good evening here in California. So, I'm here to present some of the analysis that we did. So, the flow of the conversation is quite interesting. We are trying to apply whole genome sequencing for One Health pathogens. And then, so that's Salmonella, Campylobacter, Listeria. And I expect to bring back the technology as well as the bioinformatic capabilities in the Philippines when I finish, hopefully, my PhD next year. During the onset of the pandemic, we started analyzing the whole genome sequences. And one of the emerging challenges we found is that when you have 40,000 sequences, how do you analyze that? So that will be the flow. How do you integrate? Because this is the first time that in a pandemic, we have real-time whole genome sequencing that is occurring. And that is enabling us to draw insight using bioinformatics, machine learning, and other techniques, especially epidemic modeling, in trying to draw insight from the whole genome sequences. So during an infectious disease outbreak, we usually try to identify the index case using contact tracing. But in certain instances where contact tracing is inadequate, there will be undetected transmission. This is where whole genome sequencing shines because you can use the mutation. So the RNA virus of the SARS coronavirus, it undergoes mutation. So approximately 2 mutations per month is the average. We calculated that mutation rate. And it enables you to trace. So if you have the dates and if you have the mutations, you can deduce the probability of infection. And you will know that who infected whom using whole genome sequencing data. So you use a reference to be able to deduce that mutation. So the reference we are using is the original Wuhan isolate. And for a so you calibrate it with the mutations and you can estimate. Now, for the Philippines, we have very few sequences. So I have not included the Philippine, but RITM, as well as the Philippine Genome Center, has sequenced a few. But hopefully we can add more so that we can draw insight. So what information can we get if we analyze the whole genome sequences of the coronavirus that's causing the pandemic? For one, we will be able to isolate. So if we start looking at the epidemic data that we're seeing, it's an aggregate of the cases. But if we do whole genome sequencing, we can divide the cases into clusters, the infection clusters, and actually that's what we did. We use the phylogroups as proxy for the infection clusters, use the frequencies for each phylogroup, and then we try to compute the superspreading metric. And based on these methods that we started using, we're able to see that the dispersion parameter, K. So everybody's talking about R, or the reproductive number. There's another metric in epidemiology which measures the super spreading event, which is the dispersion parameter, K. And we realized that based on the data generated, there's really what's driving the epidemic, the pandemic, are the super spreading events. Then we try to quantify the dominance using the Gini index. Gini index is used in economics to show disparity in wealth. But we want to quantify. So we are developing new quantitative methods, because in phylogenetic analysis, once you use the sequences, the number or the frequency for each particular clade does not add additional information. So we develop new quantitative methods to express this. And this, one of the methods we use is the Gini index to measure the inequality. So in UK, where UK is interesting as an example because they are the ones with the most number of sequences. So UK mobilized all the laboratories, even academic laboratories, to do sequencing so that they will be able to sequence as much, and they started sequencing 10,000, and last time I checked, it's beyond 50,000 sequences. So it's the most number of sequences is coming from the UK. And apparently the Lorentz, the Gini index is 0.8. So if you have perfect inequality, the Gini index is 1. If you have perfect equality, the Gini index would be 0. So it's 0.8 is nearer to the perfect inequality value. And that dominance of specific lineages is seen throughout countries. So I tried computing the Gini index for Australia, Spain, Netherlands, China, and Denmark. And it also shows that there are dominant lineages that predominate the pandemic, as well as in Portugal and as well as in Iceland. So we ask a question: are there mutations that are driving the dominance? So if there are lineages that dominate, what are the possible explanation? What are the causes for the dominance? And one of the popular hypotheses is the mutation of the 614 of the spike protein. There are in vitro studies that were conducted showing that this increases the infectivity in cell culture. And if you use aggregate statistics, they are dominating. So once this mutation happened, if you look at the aggregate as statistics, it shows that they are dominating. So we tested the hypothesis whether this mutation is dominating and in what particular mechanistic way. So we computed the SNP-specific reproductive number. We found out that there are indeed differences in the SNPs if you compute the reproductive number, which is the average number of secondary cases from the initial case. So that is used in epidemiology, and we use it using allelic frequencies to compute the reproductive number. And you can see the effect of lockdown. The value of the reproductive number using SNP went down to 1, meaning that they are able to control it. So the genetic data is effectively capturing the intervention. So this is also a novel finding that we have, that if you have sequenced enough, you would be able to capture the effectivity of the intervention. And this is universal for other countries also that we check. So but there's a time delay to that. And this is associated with delays in testing and detection plus the incubation period. So if you factor that delay, so we use Apple mobility data. So upon the imposition of lockdown in the UK, there's decrease in mobility, which is impacting the reproductive number. But you wouldn't immediately capture this, because even if you impose hard lockdown today, the cases will not increase tomorrow because of the incubation period. So if you factor in the incubation period as well as the diagnostic delays, you will have an elegant manifestation of the data. And then we applied, we parameterized an epidemic model, SEIR model. So we determined this reproductive number for each SNP, used that to parameterize the model. And this is the model that we developed. So the difference in the reproductive numbers able to capture the dynamics, the disparity of the G mutation versus the D mutation in the spike protein. So this is quite an interesting finding. So this model is useful because it cancels out all the effects. So the lockdown, the contact. So assuming that there's equal homogeneous contact, G mutation would overtake the frequency of the D mutation. So there's some indication that the in vitro studies is similar to the findings. So it does not mean that it's more, it's deadlier, but it infects more. So the value of the reproductive number has nothing to do with case fatality. It's just the number of transmission events. So that's all for my presentation. Thank you for listening, and we'll go now to the questions.
Thank you very much, Dr. Bandoy.